And we are live. Welcome everyone to the second installment in our webinar series called Oxygen in Cell Biology that we at Milmedtek are hosting together with the lovely folks over at Baker Ruskin. Joining us today will be Sofie Mohlin from Lund University with her lecture The Role of Oxygen and Hypoxia-Inducible Factors in Normal and Tumor Development. We are very happy to have her and look forward to hearing more about her research. Before that though, I will just take a few minutes to present today's agenda as well as presenting my own company here in Sweden, Milmedtek AB. So here is today's agenda then. After my introduction, the idea is that Krista Rantanen is going to join us. However, it seems that we are having some technical difficulties here, so we'll improvise around that a little bit. So I will just present Baker Ruskin real quick after that as well. And after that then, we will get into the main event for today, Sofie Mohlin's lecture, which is going to last for 45 minutes. After Sofie's lecture, we're going to have a live Q&A session as well. So please save your questions for that. And finishing off today's event, we're going to announce the next presenter for our webinar that comes in September after the summer holidays. So the Q&A session works like this, that you can write your questions in the chat window or chat pod in the lower right window. We can then upgrade questions and bring them up onto the main screen after Sofie's lecture. So everyone can read your question as Sofie is answering it, hopefully. And I would also like to inform you all that this event is being recorded. So this event will then be available on YouTube and at our website later on as well. So Milmedtek AB is a laboratory technology company in Sweden. And our slogan is then, translated to English, tailored laboratory and industry technology. We're in the south, outside Karlskrona, for those of you in Sweden. And we supply, install, and maintain simple laboratory equipment such as microscopes to more advanced laboratory equipment such as Baker Ruskin's workstations, for example. We are also the general agent to several leading manufacturers worldwide, including, of course, Baker Ruskin, but not limited to. I would also like to use these few minutes to present our featured product, Baker Ruskin's newest hypoxia system. This is a smaller system than the classical workstations we're used to, and comes with quite a lot of benefits that I'm not going to be able to cover right now. But the two main points that I see them is that this system will allow you to create an external environment within your workstation. So why settle growing your cells at 5% oxygen and when you can also have some cells at 3% oxygen? It also allows you to transform your traditional carbon dioxide incubator into a trigas incubator, where you can place this chamber you see on the screen in your incubator, and that will allow you to gain control over oxygen as well. So we're very excited about this, and if you want to know more and you're in Sweden, please visit our website. And if you're outside Sweden, then of course, please visit Baker Ruskin's website. I'd also like to inform you all that Baker Ruskin is going to hold a webinar featuring this product as well. So keep an eye out for that on their social media. Sadly, we're having a few issues here, so Krista won't be able to join us today. But Krista is the Director of Scientific Applications at Baker Ruskin. And Baker Ruskin is then part of the Baker Corporation as well. And together they supply both microbiological safety benches and the market's leading anaerobic and hypoxia and physoxia workstations. Baker Ruskin introduced the first hypoxia workstation, and Baker introduced the first biological safety cabinet. So long in the field and experts in the field. So if you're in the market for any of these kinds of products, please look them up. And they're also very active on social media, and please find them there. All right, so we're gonna skip ahead of schedule a little bit then. And I now have the privilege to present Sofie Mohlin from Lund University. And she's gonna present her research for all of us, and we're very excited about that. Don't forget the Q&A. So see you later.
Okay, so I will start. I'm gonna share my screen. Okay, so I hope you are able to see it as it should, and I'm sure Daniel will shout if not. So thank you so much for the invitation to present our work here today. And my lab works at understanding where the roads of normal and tumor development converge. And so what happens during embryonic development that leads to tumor formation. And today I will focus on the part of our work that looks into the role of oxygen and hypoxia-inducible factors. So we are working mainly on the childhood tumor form neuroblastoma, and we're also working on PARAGANGLOMA and PHEOCHROMOCYTOMA that are mainly adult tumors. And we know quite a lot about these tumors and the genetics around them, but we still do not know where they originate from. So just very brief background on these tumor forms. So neuroblastoma is a tumor of infancy. Most patients are diagnosed before the age of 5. And in Sweden, it actually has the lowest survival rates of all childhood tumor forms. The tumors form along the sympathetic ganglia, most commonly in the abdomen, and in there, the adrenal gland. So moving on to paraganglioma and pheochromocytoma, and short for that, PPGL, these are, as I said, mainly adult tumors, but there are still around 10 to 20% of the tumors that are pediatric. And here it ranges from 0 to 18 years old. So it's all through adolescence. And these tumors are very slow growing. They usually grow for 30 years before they are detected. And they are usually benign. But there are cases that do present with aggressive tumors and metastatic disease. And for these patients, there are no treatment or cure. So these tumors are neuroendocrine tumors, which means that they arise along the sympathetic but also parasympathetic ganglia. And also here, one of the most common sites is the adrenal medulla. So as I said, these arise in the sympathetic and parasympathetic ganglia from the sympathetic nervous system, and these in turn arise from the neural crest. And I will shortly come back to what the neural crest is. But I just want to highlight that, at least for neuroblastoma, we know that a high tumor cell expression of neural crest genes, which means that the tumor is undifferentiated. It has a similar phenotype to neural crest, relates to or predicts poor outcome. So the neural crest, for those of you who don't know, is a transient structure during vertebrate embryogenesis. And it forms as the embryo grows very early on, as the neural tube closes, and I will show you a picture on that. And there are 4 different derivatives of the neural crest. So the cranial, vagal, trunk, and sacral, and as you can see, depends on the anterior to posterior position along the embryonic body. And the larger part is constituted by the trunk neural crest. And this is what we are interested in, since this is where the sympathetic and parasympathetic ganglia derives from. So here you can see a transversal section of an embryo. And here the blue that you just see half of right now is the neural tube. And the neural crest cells are located here in the dorsal part of the neural tube until a specific time point in the embryonic development from where they start to delaminate from the tube and migrate. And depending on the different routes they take, so they can migrate dorsally or ventrally, and depending on which cues they account or meet, they differentiate and commit to different cell lineages, including pigment cell, Schwann cell precursor cells, neurons in the sympathetic ganglia and parasympathetic ganglia, and then cells of the adrenal gland, which mostly is then, of course, the chromaffin cells. So this seems pretty simple, and we tend to look upon neural crest as having the cells migrating, as I said, committing to, and if they migrate ventrally, they commit to a sympathadrenal progenitor cell, which in turns commit to either a chromaffin lineage or the sympathetic neuronal lineage, and that's it. However, as you might understand, both from the slide where I'm going and from just thinking about biology, it's not very simple. So there are a couple of recent single-cell RNA-seq papers that show that it's definitely not this simple as we have sort of thought about it. So this is from a paper from Igor Adameko's lab that was published very recently, and they do single-cell RNA sequencing of human fetal adrenal gland. And as you can see, indeed, the neural crest cell can differentiate into sympathoblasts, but both intraadrenal and extraadrenal. It also differentiates into Schwann cell precursors that in turn differentiate into chromaffin cells. So we have this intermediate step. However, the Schwann cell precursor can also differentiate or transition into sympathoblasts that in turn, immature sympathoblasts that in turn differentiate into an intraadrenal sympathoblast, but not the extraadrenal. And if that was not enough, there is also a transition phase in both directions between the intraadrenal sympathoblasts and the chromaffin cells. So you see that there are so much that we don't know about the neural crest, the different ways the neural crest go and take and how they commit. And of course, if we don't know that, then we will definitely not know where tumors form. So it's definitely very important to look into and understand normal development to understand also tumor development. So there was another paper from Frank Westermann's lab in the same issue, Nature Genetics, where they do also single-cell RNA sequencing of fetal adrenal gland. This is from mouse though, but they find 21 different cell subtypes. So these are not all of them even, but we have the Schwann cell precursors, what we call the bridge cells, when they start to commit, the connecting cells, and then out to differentiated chromaffin and late chromaffin cells, and neuroblasts and late neuroblasts, so differentiated neuroblasts. So again, our question is when, how, where do any of these cells transform into a tumor cell, and what is the actual cell of origin, or as I tend to and like to stress that it's rather cells of origin. I am quite sure that there's not just one cell of origin for all of these tumor subtypes. So today we are of course focusing on oxygen and hypoxia. So this is what I will focus on from our research perspective as well. And I'm sure that very many of you are familiar with oxygen and hypoxia and how it works, but I will still go through it a little bit from and apply it to what we are working on. So as you know, oxygen is one of the most important environmental factors during development. And we also know that oxygen levels fluctuate as the embryo develops. One of the factors that the cell initiates or induces to respond to these fluctuating oxygen levels is the hypoxia-inducible factor 2 alpha. As you know, there are 3 different isoforms, HIF1 alpha, 2 alpha, and 3 alpha. But today I will only be focusing on HIF2 alpha. So normally HIFs and HIF2 alpha as well is regulated oxygen dependently. So this is the structure of HIF1 alpha and HIF2 alpha. I'm sure you all recognize them. And at normoxia, what we call normoxia, when there's oxygen present, this HIF alpha protein is hydroxylated at these specific proline residues. When it is, the von Hippel-Lindau protein binds, and this leads to proteasomal degradation of the HIF alpha protein. However, if the protein still stabilizes and translocates into the nucleus, it's hydroxylated at another site, an asparagine residue, and then it's targeted by the factor inhibiting HIF. And this in turn hinders formation of the complex needed to initiate transcription. At hypoxia, however, there's no oxygen, hydroxylation cannot take place. The HIFs move into the nucleus, binds to its binding partner ARNT and transcriptional cofactors such as CBP and P300. So this is the simple way. You're all aware of this, and it's of course the simple canonical way of looking at it. So I've made a schematic that is extremely simple and presents this with 2 arrows sort of. So close to blood vessels, there is high oxygen. Cells don't express HIF2 alpha, but the further away from the blood vessel we go, the less oxygen has diffused and reaches this site, and the cells start to express HIF2 alpha. And this, I'm going to come back to this sort of setting with oxygen and blood vessels and HIF2. So that's why I have this slide now, so you will recognize it later. So we know for sure that HIF1 alpha and 2 alpha are regulated post-translationally. And this is from a very old paper, but it quite nicely demonstrates that HIF1 alpha is expressed very early on. It responds to the acute phase of hypoxia, after which it slowly starts to decrease in expression, whereas HIF2 is rather increasing with time, and we usually say that it's responsible for the prolonged response to hypoxia. This is, of course, very also simplified, but still. However, it's been sort of noticed now that it's not only this easy. And that's the post-translational regulation is what I showed you in the picture above and how we all talk about it. But we actually know there's been several papers over the last couple of years that show that HIF2 is regulated transcriptionally as well. And you can see that with time, the protein expression increases as well as the mRNA increase, that the mRNA increases, whereas this is definitely not seen for HIF1. What kind of impact this has is not really clear, but it is definitely clear that the 2 factors, HIF1 and HIF2, are regulated differentially. So we've been talking about oxygen-dependent regulation, but we also know that there is oxygen-independent regulation. And these are just a summary of that. I think it's pretty nice. So there is, for example, oxygen-independent modulators of degradation of the protein, but also modulators of translation. And I'll come back to this, touch upon this very briefly later on as well. And one of the ways that this can happen is via signaling pathways, including the PI3 kinase pathway. And PI3 kinase is also something that will come back in my presentation from our work. And here, this is just to show that at oxygen levels where, so 5% oxygen, whereas, we have oxygen, HIF2 should not be expressed, but it is. And when we inhibit the PI3 kinase pathway using this conventional LY inhibitor, we completely ablate HIF2 expression. So why do we study hypoxia and oxygen in the particular tumor forms that I mentioned and presented in the beginning? And why do we focus on HIF2? I think that would be pretty clear, and that I convinced you that that's interesting after these 2 slides. So paraganglioma and PHEOCHROMOCYTOMA are the only tumor forms where it has been detected mutations in the HIF-2 alpha protein itself. So paraganglioma and PHEOCHROMOCYTOMAs are driven, around 80% of the tumors are driven by genetic changes. And there are many of them that lead to a pseudohypoxic phenotype, and these tumor or these patient group presents with a worse prognosis. And within this subgroup, there is the patients that have the mutations in the HIF-2 protein to create the pseudohypoxic phenotype. The mutations lead to stabilization of the protein and increased half-life, which of course then or end up with increased activity. Creates the pseudohypoxic phenotype, and within this group, this correlates to even worse prognosis. And as you remember, I said that most tumors are benign for sure, but there are also these patients that we cannot cure, and these patients are the ones with this pseudohypoxic phenotype mostly. In neuroblastoma, we have not seen any such mutations in the HIF-2 protein itself, but we have seen this. So located very close to blood vessels, which means that they have access to oxygen, are cells that do express HIF-2 alpha very intensely, also creating this pseudohypoxic phenotype. And it's been shown that this correlates to the presence of these cells. So it's not high expression, but it's intensely HIF-2 expressing cells correlate to distal metastasis as well as poor prognosis. And in particular, this is highly interesting to us because these cells are very immature and present with a neural crest-like phenotype. So I'm going to go into our data, but before that, I just want to show you a model system that we use that are called CRESTOSPHERES that we established from trunk neural crest. And we use chick embryos as our main model system in the lab. And I think that not very many of you are familiar with maybe the chick embryo and in particular CRESTOSPHERES. So just a very brief introduction to that. So this is how the embryo looks in the egg. Obviously, we have injected a dye underneath. It's usually not blue, but that's just so that you can see the embryo here. Up here is the head, and then you have the embryonic body. And this is around embryonic day 1.5. They hatch after 21 days, so this is early. So normally when we do our experiments, we have the embryos within the egg, and they continue to develop. But when we extract or establish CRESTOSPHERES, we take out the embryos and we dissect this very small part. So we do it under the microscope and dissect this very small part of the neural tube, roughly between somites 10 and 15 to really ensure that we only have the trunk neural crest. We dissociate the tissue and culture them as spheres, and you can see this very nicely shaped. So there are spheres and they derive from the trunk neural crest, so they're called trunk CRESTOSPHERES, easy as that. And this is a very nice system because we see that when we culture them over several weeks in vitro, they maintain their self-renewal capacity, so their stem cell phenotype, and they retain their multipotency. So you can see here that we can make them differentiate into a variety of different lineages: smooth muscle, neuronal, glial, and even cartilage, which is quite recently shown. We showed this and then it's been confirmed. Okay, so I'm going to go into then HIF-2 alpha that we focus on and what we are doing with or looking at HIF-2 in the neural crest. So I'm going to just start with in the chick embryo, and over the time course, we see that HIF-2 alpha mRNA is expressed, and it sort of peaks here around Hamburger-Hamilton stage, which is what we, in the chick embryo field, counts age or development in, and you don't have to understand what it is, other than it's over the course of a couple of days. So this is around 1 day and this is around 4 days. And you can see that it peaks around here, which is exactly when the neural crest cells migrate. This is mRNA. We wanted to confirm that we see it on protein levels, so we did an IP, and indeed we see HIF-2 protein. And we also looked at HIF2 expression between CRESTOSPHERES, so we established them from trunk that I showed you and also from cranial, and there is an enrichment of HIF2 in the trunk as compared to the cranial neural crest. So we know that HIF2 is expressed in the chick embryos. So we also wanted to look then at the actual oxygen levels. And so we did measurements, and these were all performed by Emma Hammarlund here in Lund. And in our paper that I will present some data from, we measured oxygen directly within the neural tube at the trunk axial level. So you can very beautiful see here at Emma's setup. Here, down here is the embryo that she's taken out, and she cultured it ex ovo. So she takes it out from the egg, keep it alive, and then directly measure oxygen. And you can see here on the picture very nicely that the microsensor is within the tube. And we can see that at very early time points, at around day 1, we have very high oxygen levels. They decrease over time, some kind of peak here at around stage 18, again, when the cells migrate, and then down to very low levels. Emma and her group very recently published another paper where they looked even more into this. So here they measured oxygen at other places, so the sacral neural crest at the very end, so at the very end part of the tail of the embryo, the vagal, which is just above the trunk, in the head, and in the heart itself. And you can see at all these different locations in the embryo, we see the same pattern. And Chris also performed Western blot looking at HIF1 and HIF2, both within the embryo as well as in the allantois. And you can see that in the embryo, expression of both proteins increase with time, whereas here it decreases in the surrounding. So we know how the oxygen levels look. We know that we have HIF2, but we also wanted to look spatially and at protein level again. So we stained sections from embryos of 3 different species, human, mouse, and avian. And this is around embryonic week 6, so it's very early of a human embryo, and the chick embryo is at a stage that corresponds to that, whereas the mouse embryo is a little bit older. So here you can see that HIF2 positive cells are migrating. These are the trunk neural crest. They have left the tube, which is exactly what we expect at this time point, and they are migrating towards the sympathetic ganglia. And you can see the same here in the chick embryo, and you can see that they collect in the sympathetic ganglia already here. And this is what we see in the mouse. So we don't really see very many positive cells migrating, which is because this embryo is older, but instead we find them in the sympathetic ganglia. So we do have a rationale for study HIF2 alpha in the embryo. So we started by knocking down HIF2 alpha in these early embryos. We inject DNA into the neural tube when the embryos are within the egg, and we electroporate the embryos to have the cells take up the constructs. So to begin with, we again dissected the trunk part of the neural tube, and we ran RNA-seq on embryos that had been injected with a control construct or a knockdown of HIF2. And I'm showing you the top 10 up and down regulated genes in case you recognize any of them. I'd be happy to discuss. And when we do gene set enrichment analysis, we find that there are enrichment of genes associated with migration, embryo growth, which makes sense, invasion, epithelial to mesenchymal transition, also makes sense, these are migrating, and tumor growth. So RNA-seq is great, but of course we need to look into biology as well. So we did the same experiment, but had the embryos continue to grow. And we didn't really look for the embryonic phenotype, but we started to notice that the embryos where we knocked down HIF2 were smaller. So we quantified the developmental age of the embryos, and indeed when we count somites, which is then a measure of the developmental age, we see that when we knock down HIF2 with a morpholino, the embryos are significantly smaller, and we see this the same phenotype when we knock out HIF2 with 2 different CRISPR constructs. So we now know that the embryo is affected, somehow. So we wanted then to go deeper into the neural crest specifically. So we sectioned embryos that we had manipulated, and we stained for HNK1, which in this setting is a marker of migrating neural crest cells. And this is, again, just want to stress that it's at the trunk level, of course. So here you can see that we have done one-sided electroporation, which means that we electroporate, we have current across the embryo so that only the cells on this side take up the construct. So the green cells are the ones with our CRISPR construct, and this is a control. When we stain for HNK1, you can see that it's expressed on both the control side and the electroporated side. So there's no difference within the internal control and the electroporated side. However, when we electroporate with a CRISPR construct knocking out HIF2, you can see in the control side the very beautiful HNK1 migrating cells, whereas these are basically absent in the electroporated side. So what goes hand in hand with migration is, of course, proliferation. So we did real-time live EDU incorporations assay. And this is from the specific control Morpholino. And you can see now the targeted cells in red and the proliferating cells in green. And when we look at the embryos where we knock down HIF2, there is almost only or a very high proportion of proliferating cells. And when we quantify this, we see a massive increase in proliferation when we knock down HIF2. And we also wanted to look at self-renewal, so their stemness capacity. So we use now our CRESTOSPHERES cultures. And to do this, we established, or we knocked down, used the CRISPR constructs with control or knockout of HIF2 in the embryo. And from these embryos, we established the CRESTOSPHERES cultures. So the CRESTOSPHERES, the cells, were already manipulated when they started to grow. And indeed, when we quantify the number of spheres as well as measure their size, there's a huge increase when we knock out HIF2. So I've been telling you what happens when we knock out HIF2, but of course we need to know, well, then what happens when we overexpress it. So again, we use an overexpression construct and we get the same phenotype. So the embryos are smaller, they start to proliferate massively, and we abrupt migration. So we conclude that it's not that HIF2 should be very high or very low, that rather it needs to be very balanced if the development is to be correctly or to be correct. So in this, going back to the RNA-seq data, I'm not going to go into it, but we looked for enrichment of different signaling pathways, and there were actually very few, but one of them was the PI3 kinase family. And that's, of course, interesting because what I told you before, and also because we have published 2 papers where, first of all, we show that PI3 kinase, and specifically via mTOR complex 2, regulates transcription, so the sort of non-canonical regulation of HIF2, and also that when we use a novel inhibitor of inhibiting PI3 kinase together with mTOR and PIM, we see a great effect towards tumor growth in neuroblastoma. So this is, of course, quite intriguing. So just to summarize this part, we show that when HIF2 alpha levels are skewed in the neural crest cells, so we target the neural crest cells specifically, it's not the whole embryo. We can see that the embryogenesis is delayed. We see enhanced self-renewal. We alter the migration, maybe cell fate. We see an induced proliferation, and I didn't show you that. I will in a while, that we not only do we have this global change in expression, of course, from the RNA-seq, but we also looked more closely into trunk neural crest-related genes, and also there we have a huge effect. And the question now is, of course, how does this relate to tumor genesis and tumor initiation? And this is something that we are working on. Now I'm going to go over to something that I call non-canonical regulation. And I know that that's few of you that probably don't like that, but it's just, you know, to show that we are now talking about something that is not the usual way we look at oxygen and HIFs. And so again, you see in this slide, as I said, it would come back, so the canonical way. But we also know from a couple of papers now that HIF-2α is expressed in these perivascular niches. So this is from a patient specimen, and when we do patient-derived xenografts, so we have small pieces of tissue from tumor tissue from a patient, implant into mice, take out the tumor, we also find these HIF-2α positive perivascular cells. And again, this leads to a pseudohypoxic phenotype. And what I changed here is then that we have HIF-2α expression independently of the oxygen level. And normally we look upon HIF-2 as being translocated into the nucleus to act as a transcription factor. And for sure, that's true. But it has also become evident during the recent years that HIF-2α is not only expressed in the nucleus. So here, for example, these are patient-derived xenograft cell lines, and you can see that at all 3 oxygen levels that we look at, 21, that is, you know, the lab, what we use in the lab, 5% what we say more is a physiological oxygen concentration, and 1% to but there's still a significant fraction of the HIF-2α protein that localizes to the cytoplasm. At 21%, it's almost exclusively in the cytoplasm. At 5 and 1%, a large fraction of the protein has translocated to the nucleus as expected, but there's still definitely protein left in the cytoplasm. And we looked at this spatially and in single cells in our human embryos, and indeed we find cells that express HIF-2α in the nucleus and the cytoplasm, and also cells that only express it in the cytoplasm. And this paper is from almost 10 years ago now, and I'm showing you because it has these cap-dependent translation that they look into. And in here they see that HIF-2α forms a complex together with RBM4 and other binding factors or cofactors, and they localize to the polysomes to initiate translation. So this is not transcription, it's not in the nucleus, it's in polysomes, and they initiate translation of a large set of proteins, but also a distinct set of proteins, including HIF-2 itself, actually. So we are now interested in looking into this. So what are these non-canonical functions of HIF-2 and how does this relate to our results? So we have used PT2385. I will now say PT, it's easier. And this is the first HIF-2 specific inhibitor that's been developed. And the PT binds to a pocket that is only present, or the structure of this pocket is specific, or it's very different between HIF-1 and HIF-2, so that's why PT can only bind to this pocket in HIF-2. And when it does, it's a sterical hindrance from HIF-2 to bind to ARNT. So we looked into the chick embryo, and just to remind you, when we knock out HIF-2, we see this phenotype that the embryos are younger, they develop at a slower pace. We do not see this when we treat with PT. And this is what I told you in the summary, but I didn't show you. So we look at these trunk-neural crest specific genes. So these are enriched in the trunk-neural crest. And as you can see, when we knock out HIF-2, we have a huge increase in most, or decrease, of course, sorry, in most of these genes. When we treat the embryos with PT, there is no difference in gene expression. So we want to tend to look in our PDX cells and at the expression of these trunk neural crest specific genes. And here, just like in the embryo, there is no change in expression of these genes at all when we treat with PT. So we confirmed that our inhibitor works. You can see here in the control, the DMSO treated, we have a very nice immunoprecipitation showing that HIF-2 and ART binds, where when we treat with PT, this complex formation is completely ablated. And we also tried our inhibitor, the PT, on renal cell carcinoma cell lines that we know are driven by HIF2, and also the HEP3B liver carcinoma cell line, and it works perfectly as expected. So we looked at the expression of HIF-driven genes, and we know that the inhibitor works, but we don't have an effect on these genes. So VEGF and BHLHE40, which is more commonly known as DEC1. So to assure that in these cells HIF2 is actually driving these genes, we knock down HIF2 alpha using 3 different siRNAs, and you can very nicely see that we have a good knockdown of HIF2, and indeed we have a very nicely decrease in VEGF and DEC1. So HIF2 is actually, we know it's really driving VEGF and DEC1 as we would expect, but when we inhibit the binding of HIF2 alpha to ARNT, we don't see an effect. So we went back to our chick embryo and we looked at these HIF downstream targets when we treat with PT, no effect, just like we see in the patient-derived cells. So again, also here to verify that to driving genes in the setting, we knock out HIF2, we used the CRISPR construct, and although there's no huge effect on VEGF, which could of course be a compensatory mechanism for HIF1, for example, but when we look at the HIF2 specific target gene DEC1, there's a huge effect. So again here, HIF2 is driving downstream target genes, but we don't see an effect when we inhibit the complex formation. So as you can see, a lot of this data is unpublished. We are preparing a manuscript for some of the data. And I'd be happy to take questions and discussions. There's a lot of this that we don't know. And of course, a lot of these results were and are still surprising. So to conclude the first part, the first part, when we look at knocking out HIF2 alpha in trunk node CREST cells, we see an effect on migration, proliferation, self-renewal, gene expression, and developmental growth. But this is from an expression level view. So what we do is we knock out or overexpress HIF2, and then we see these effects. But the second part that I talked about is that we also have HIF2 in the cytoplasm that we don't really know what it does, and especially not during normal development, embryogenesis. It is obviously ARNT independent mechanisms that drive expression of downstream target genes. We cannot say that HIF2, that it's not via transcriptional effects, right? We only know that it's not via the binding of HIF2 to ARNT. And obviously it's protein dependent because when we knock out HIF2, we do see these effects. So the question now is, what role does the localization play? Is there non-transcriptional functions? And how does this relate to tumor genesis? And these are, of course, the questions that we are working on trying to answer. So there are many people to thank, and all our collaborators, and of course, most the lab doing the work that I'm very grateful for a fantastic group. And at last but not least, the funding agencies, of course, making it possible to do this work. So I'll be very happy to take any questions and let me see, discussions.
Thank you so much, Sofie, for that fantastic lecture. I'm so glad we managed to bring you on.
Thanks.
Yeah, everyone, please, if you have any questions relating to Sofie's fantastic lecture here, then feel free to write that in the chat window and we'll bring them up on the screen. And as we're waiting for that, I'd also like to remind everyone that in the invitation you got, there's a link to our website where you can find a few of relevant articles relating to Sofie's research as well for a couple of days. So we're looking forward to
Everything is crystal clear. I'd be happy to take any suggestions or the discussions as well about the PT results and why we don't see the effects in neuroblastoma cells and during embryogenesis that we do in other tumor cells or tumor forms. If anyone has thoughts about that.
It's starting to seem like everything was extremely clear, Sofie.
Yeah.
But I assume that if any one of our attendees here have any questions coming up later, they're free to contact you.
Oh, more than happy to for that. Absolutely.
Okay. All right then. With that, then we will present our next webinar as well, finishing up this event. So let me find that there. So in September, we have the privilege of bringing on Emma Hammarlund, also from Lund University, for our third installment of this webinar series. Her title of the lecture is going to be Living with 21% Oxygen Was Never Easy. Hypoxia as the Norm in Animal and Earth History. Save the date, the 30th of September at 3 o'clock Swedish time. So convert that to your time if you're interested, and keep an eye out for invitations or keep an eye out on Baker Ruskin's social media in the future. Of course, if we're the Swedish partner of Baker Ruskin, and we try to highlight Swedish research. But if any of you in the audience here today in Swedish research are interested in a similar event, such as the one we've hosted today, we're more than happy to discuss and setting up a future webinar, highlighting your research and allowing you to network with both national and international colleagues. And finally, I will then like to say thank you all so much for attending, and I hope to see you again next time. And again, a big shout out to Sofie and her fantastic lecture. Thank you so much for attending. Bye all. See you next time.